I have a couple of minutes of background, and folks who participated in the first call might this might sound familiar for them, but it's important that I cover the background and set the table for today's conversation, so we can have it be as meaningful as possible. And I do want to emphasize that I have this a bit of opening remarks, but again, people who were on the first call know that it became a very lively conversation, and so we have resource people, which I'll introduce here in a few minutes, but we have everyone on the call who should also have equal time to share a comment or a question, or two or three. So I guess I would just like to welcome everyone to the call. For those that don't know me, my name is Matt Kleinhens, and I am a vegetable extension specialist at Ohio State University. I work from an office at the Ohio Agricultural Research and Development Center in Worcester, which is where I am now. And my job over the next 90 minutes is to make sure that you are repaid for the time that you spend on this call, repaid in different ways, but primarily with information that you can use, and also repaid with the knowledge that your input is very important to us and to the group, and that you're very much welcome to join us going forward as we work to help everyone get more from these important products, crop biostimulants that have important characteristics. And just to set the stage further, because this issue continues to emerge, if you will, continues to show itself, I'd like to emphasize the characteristics of the products that we do intend to discuss today and going forward. First characteristic is that they contain microbes as an active ingredient. That's essential. We are excluding products that lack microbes as an active ingredient. The second characteristic is that they are OMRI listed, and that they have the potential to be used by sustainable organic growers, especially vegetable growers. The third characteristic is that they are advertised to enhance crop growth and or resistance to stress. Now, this should be their primary function. It may not be their only function, as described by product literature and so on, but it needs to be their primary function as opposed to biocontrol-like activity as a biopesticide. And the fourth and final characteristic of these products is that they are applied to vegetable crops. So with due respect to the people who work with products that would be applied to agronomic and other crops, and maybe even involved in animal production, we are so far excluding them from this particular conversation, focusing instead on ones that are used on vegetable crops. So before we get down to our task at hand this afternoon, I'd like to cover three other important bits of background. There are just a couple of ground rules for the call. I'd like to emphasize the need to mute your phone when you are not speaking, especially if you happen to be in an area that has some noise in the background. We'd like to avoid an embarrassing or disruptive moment. Just as a matter of courtesy to others on the call, you might be in an area where there's some noise, so keep your phone muted unless you intend to speak up, but obviously don't let the mute be on the whole time unless you truly want it to be. Do speak up. Secondly, listen carefully, as I mentioned, but speak up, and we have a panel of resource people, but we definitely need to hear your voice as well. I'll also emphasize that we have an email address. There is an email address that you can send comments or questions to directly, and those emails will be shared with the project team, some of whom are on the call today, myself and Bonnie Only as two representatives. So that email address is vege, V-E-G-E, bio, B-I-O, stims, S-T-I-M-S, ferts, F-E-R-T-S, @gmail.com. So again, that's vegbiostims @gmail.com. And if you have any questions about that, just contact me afterward. The reason for that single email is that with that email, as we get going in this work, you will have the opportunity to essentially reach any number of people, myself, Bonnie, others, with your question or comment directly, as opposed to...
Andy Radin, University of Rhode Island.
Hi, Andy. We're just getting started, and I'm just reviewing the ground rules for the call, which you are probably familiar with. And I just mentioned the email address. So are there any questions about what I've said so far before we get further into this background, and I need to turn it over so others can speak, but I do need to go through this background. Okay, so if we don't have any questions, we can continue on with it. It's important to recognize the support that's made these calls and the work that we're engaging in possible, and that support is coming from the USDA Organic Transitions Program, the North Central Region SARE Research and Education Program, and companies and organizations represented by everyone on this call, and the universities too. So it's we really appreciate that support, and it's catalyzing this, making this conversation possible. Third bit of background. You might be wondering who else is on the call with you. So some people patched in early and they heard some of the chatter. Policies, do not allow me to share exact contact info with individuals. But what I can assure you is that there are people on the call that represent 10 states, coast to coast and north to south, different universities, and different biostimulant manufacturers and different individual farms and consultancy businesses. Everyone is directly involved in sustainable organic vegetable production in some way. But of course, as you call in, as some people have, you're welcome to introduce yourself, and as you speak up, do the same so others can know who you are. So that's the background. And we're going to focus on two main topics today. What research says about the performance of these products on sustainable organic vegetable farms. And we'd also like to include summary, so-called bottom line or take-home messages learned through the many on-farm tests the people on this call are familiar with. Those are our two main topics for today. But before we turn to those, I'd like to offer as a bit of a warm-up, a brief recap of what we discussed on January 24th, especially for folks who weren't here with us then and have not had a chance to listen to the call. So during that call, we defined pretty clearly and fairly comprehensively what these biostimulants are and what they are not. We also offered some advice on how to think of them within the context of typical farming operations and other inputs. Second, we focused on product labels and extracting information from labels, because as you know, these labels can be overwhelming with the information they contain, or actually the other way too, somewhat underwhelming in the information they lack. The third topic we covered on January 24th was how these biostimulants are supposed to work. So we talked about mode of action and touched on the 4 or 5 different ways that they have been demonstrated to work. And the final topic we covered on January 24th was basically, do they have a role in sustainable organic veg production? We asked for basically a thumbs up, thumbs down vote from people on the call. Now, you can verify for yourself, but I recall the vote was unanimous, thumbs up. But there was some hesitation, because it's been clear that it's difficult to get value from applying these products under many circumstances consistently. So that brings us to today, and the topics that I mentioned up front, which is, what does the research say, and what are the bottom line take-home messages that can be learned from the on-farm tests. And all of us can add to that conversation for sure, and I'm looking forward to hearing from you. I learned a lot from the first call, and I want to thank the people who made that possible. But here we are today talking about the research side of things, especially the on-farm research that's been done. So we have 4 people on the call that are committed to helping us through with their input, but everyone else is invited to as well. I'll introduce who our resource people are, and then I'll kick the conversation off with just a question to one of them. So we have Mr. Steve Becker and Dr. Dennis Warnke from Tainio Biologicals, and I apologize if I am mispronouncing that. How is it pronounced?
It is pronounced Warnke.
Okay. We have Dr. Jozsef Racsko from Mycorrhizal Applications, also a manufacturer. We have Dr. Philippe Douillet of Ecomicrobials, which is a third manufacturer. And we have Dr. Jung Wong from OARDC. Steve, Dennis, Joseph, and Philippe have a lot of experience evaluating products with growers. And Jung has some experience of his own with during his graduate and postgraduate work. So we can get down to business. And again, I'll just caution people, if your microphone is live, be mindful that whatever's happening there can be heard by all. So Steve and Dennis, I guess I'll pick on you first, if you will. There are a lot of university people, a number of university people on the line today. Most agree, I won't speak for them, but I would say that most agree that these biostimulants have potential, but typically they need to see data before they can recommend them. Okay? And in my experience, again, I won't speak for others, but in my experience, some data have been hard to find. So what do you suggest that these advisors tell growers about products? How do we bridge this gap that's created by a lack of publicly available information? Surely companies do have data, but it's generally not in the public domain, so to speak. So we'll go around the horn. We'll hear from Steve, Dennis, Jozsef, and Philippe on this. But Steve and Dennis, if you want to chime in first, that would be great.
Yeah, this is Dennis, and the way I look at it is, we talked a little bit about last week is, a lot of companies have been out there doing biologicals for over 30 years. And I think that it has changed now where it is becoming more accepted, and there's starting to be more university information out there to back a little bit about what biology can actually do within that soil environment. And so I think as time moves forward, a lot more research and information will be made available now. So many of the small companies just did not have the budget to do a lot of extensive testing, and university testing was, in some instances, was hard to come by when it came to biologicals. So I think as things move forward, that part of it, I think, will start to go away, and we'll start to see a lot of good information coming out on the testing of biology.
Okay, so
from my perspective, I agree with Dennis. I rely pretty heavily on searching for the benefits of the organisms. So a lot of times, instead of trying to see exactly what a product or a label is doing, if I see different organisms and different species on there, what I'm going to do is I'm going to search for what are those organisms doing, and usually I'm looking at it from a specific activity standpoint. So if I'm looking for nitrogen-fixing bacteria, the azotobacters, azospirillums, and some of the others, I'm going to see what crops have been studied, what sort of efficacy has been done, and we are starting to see more and more data along those lines, which is helpful. Then beyond that, what we'll see is data supplied by each company, because company to company, they're using different organisms, different concentrations of those organisms, and they have different efficacy and different testing results with those. So seeing what is available both from university as well as what the company has, what their literature represents, and what their sales force has. And a lot of times it's word of mouth, which can be fairly difficult. It's hearing from people, well, do you have testimonials? And what are people physically seeing, which doesn't necessarily hold up to the rigors of peer-reviewed data, but years and years of anecdotal can sometimes add up and give people, if not necessarily the exact data, it can give them perspective on how to use the product and how to make it work. So I agree with Dennis in that we're kind of in a transition period where more research is being done, but we still have a hurdle to overcome to truly understand what all of these different organisms are doing in our soil environment, our rhizosphere and our phyllosphere associated with plants. We're starting to see more research into induced systemic resistance. How are these organisms affecting the plant's immune system, just like we're seeing in the human microbiome? We're starting to realize and understand that these organisms play a significant role in genetic expression of humans, and we're starting to see similar responses and similar understandings associated with these organisms in our phyllosphere and our rhizosphere that we see genetic response in the plant. So it's not just a simple application, and C, we're starting to have a better understanding of shifts in phytohormone levels, shifts in protection and just systemic resistance. But again, we have a good amount of research that still needs to go into it to really create a matrix, a database where we can see, I have corn, I have Azospirillum brazilans versus Azospirillum lipotherum, which is going to give me a better nitrogen input based on my field, my conditions. And unfortunately, I haven't seen data that specific yet. It's starting to come in. So my take on it is, let's keep researching, let's keep plugging away, and we'll find more data and mine more data and have fun with it. So hopefully that helps explain a little bit of my perspective on it.
Sure, I think there's a lot of good in both of those sets of comments, and also I think a fair number of questions that could follow up. Before we turn to Philippe and Jozsef for their input, I'll just ask again, if you're not speaking, if you could mute your phone. There's quite a bit of noise in the background somewhere. I don't know if it's road noise or what, but Philippe, do you have anything to add to what Steve and Dennis have shared to this point about, where are the data? Where are the data on efficacy? What do you recommend to an extension person who needs or would prefer to have data in a refereed journal publication on efficacy, not necessarily mode of action, controlled environment studies? We're talking about efficacy on farms or in farm-like settings.
Right. Actually, most of my work is the contact with the farmers and with people that are technically doing the advice to these growers. And I have seen an evolution in the acquisition of information from the big farms, from the people more concerned about what they are doing to their soils, what is the long-term impact on their activities. One thing that we have to be very aware is that we don't look for a magic bullet. We know that everything is integrated, and it has to be done in conditions that favorize the effect of the microbes. We want to put them in conditions in which we don't apply any pesticide or anything that can come and harm what we are applying to the system. Also, you have to see that is not a single application of a compound that can hurt the production, but if you look, for example, at the effect of copper, for example, very minute amount of copper come in very many different of the ingredients they put in their blends of fertilizers and biocontrol agents, whatever. The important part is when you add them all together, what is the total concentration we are adding real to the fields? Sometimes we have seen elimination of very good results that we get in some fields in the next field just because we looked at all the stuff that has been added, and we ended up with toxic effects of this copper, for example. Yeah. Then I think people are starting to gather more information, getting more aware of working with living organisms.
Good points. So would that necessarily require, and Jozsef, I'd like to hear from you in just a minute. Philippe, would that necessarily require the grower to somewhat build the system around the use of these products? Like maybe refrain from using copper or any other material that would possibly harm the inoculant?
Well, I think that we are going into a phase. I think this is not only in agriculture, in aquaculture, where I work a lot, where the people that are successful are the people that are gathering information, that are keeping records of all these things, of all the inputs that are adding to their system, so that they, at the end of the day, they know exactly what has been put in, what comes in the water, they have a balance. Some compounds can get degraded. Some pesticides, for example, through time they get degraded. Then that's fine. It's great, actually, that they have that process. Other things like copper, no, they stay there. They get attached to the organic matter. They bound, they chelate, yeah, and they become toxic as they accumulate through time. Then I think that's why it's so important to for the people to be more aware of the whole contextual effect of what they're doing.
Perfect.
If I can add in a little bit there, an example that comes to mind of the idea of building the system around the products. If you go out and buy a nitro-coated legume, if you specifically are trying to bring rhizobium into a new field scenario, and you are applying a seed that has been coated, it's coated with those nitrosomonas, those legumiferums, all those rhizobia species. If at the same time I'm applying an antimicrobial around that seed with that seed as a side dress, I'm basically counteracting the progress I'm trying to make. So we have to give these organisms time to establish and a space to establish. So the general, in my mind, of do we need to build a system around these, I think about it as, well, yeah, if we go into a situation or a system knowing that we're doing damage, how do we fix that damage? If we go in with, like Philippe is saying, copper, for example, a lot of our fungi and a lot of our bacteria are exceptionally sensitive to those. So we do have to be especially cautious with those. We have to keep an eye on our weed species that are starting to be present associated with the copper. But yeah, I think that when we are knowingly causing damage, we have to come in and we have to fix some of that damage. And there are ways that we can do additional inoculations. We can add different foods that are specific for the bacterial and fungi and feed those populations. The example that I like to think of is if I go into the hospital and I look around, I'm going to see in an emergency room people with different amounts of need, and our soil is basically the same. We have to triage this. If I have a soil that's basically unlicensed, I have to give very simple foods, and I have to approach it from a very simple standpoint to encourage that to develop a biologically, ecologically balanced system. If,
Sure.
on the other hand, I have an organic system, an organic field that I've been using compost, high-quality compost, humates, fish, other materials that encourage and establish my biological communities, my ecological communities, I have a bit more resilience there. I can throw a more complex food base at that and know that my system is going to have a bit more resiliency. So part of this is going to be knowing what I'm doing, knowing what's been done, as Philippe was saying, and building the system so that I'm encouraging those biological components to survive. If I have a seed type that I know is supposed to be planted at a ½ inch, and I decide I'm going to plant it at 3 inches, I know that I'm going to have a less beneficial effects of it. So when we go into these, we have to have the mentality of, what am I going to be doing? Am I building the system for a biological, or am I detracting from a biological system?
Understood. I think.
We know that we're detracting. How can we boost it back up? So I definitely agree with that idea that we have to build the system.
When you and I want to hear Jozsef as well in just a second, and I certainly don't want to take the conversation in a direction that may not be as profitable, so to speak. But to me, owning my ignorance, when you walk into that emergency room, if you're able, one of the first questions they're going to ask you is, "Are you on any medications?" And they do that because they want to know what the interactions will be between medications you're on and what they may give you. And what I'm hearing now in the last 5-10 minutes is the possibility that our biopesticides and other biocontrol agents could be undermining the biostimulants. And I know that there are probably case-by-case bases where we don't have to worry about that, but is my concern exaggerated? Or is there actually something to the, hey, if you're also using some, various products to deal with your pest and disease issues, your microbial inoculants for biofertilization, biostimulant role may be worthless. So that idea of building the system around the microbials has a weak link in that farmers are often not in a position to say, well, I can avoid using copper, which is a good protected fungicide, because it for the sake of being able to have my inoculant work better. That's a conundrum.
When I talk to this is
Dennis, when I talk to a grower, and maybe to answer that a little bit, I think part of this has to come from the manufacturer making sure that with biologicals is you can't really order something online and expect a result from that online without having an understanding of that product. And so it takes a lot of communication and education, and communication on my part to find out what we're talking about is what is the grower's cultural practices, what is their history. I need a soil sample not only for nutrition but also to see where their CEC, their organic matter, their carbon to nitrogen ratio. All of this gives me information of how well biology is going to succeed within the environment that they're placing it in. And I agree with you 100%. The first thing they're going to do when you go into an emergency room is they want to know what other medication you're on so that they can prescribe a program for you to get you well. And generally they also do an examination and take a blood test. And as a grower and also as a manufacturer or a company or a rep out there, we need to make sure we're both collecting all that information so that we can give proper recommendations on how to maintain that biology within that environment. And a lot of times I tell my growers is there is going to be times that we know we are going to do things detrimental to that biology. Now, understanding that we have done that, now how do we go back and fix it? And that may be, as Steve was talking earlier, a food. It may be re-inoculation. There could be a lot of specific things that we would go into that environment with and fix the damage that we have done. And I've seen that in the golf course industry where it may be a situation in the wintertime where they come in with the fall application of a fungicide. And we realized that most everything we had done through that entire growing season, we have set back tremendously. And so what we need to do is understand in that springtime, we need to come back and get those guys functioning again. And how do we do that? And an understanding how we do that in that environment is the same whether we're talking golf course turf or agriculture or whatever. It's just making sure we have that information. And as Steve had talked about, if we're in a sand-based green, which is 90% sterile environment, we're going to have to do it differently than we are in a organic field that has 3 or 4% organic matter and soil structure. So understanding that and the grower understanding that is key to getting biology to function within that environment.
Good points. Good points. Jozsef, we're on a track here, and we've set up the call to address the take-home messages that are available from on-farm research, which I know you and others on the call have been involved with a lot. And the first question was kind of put out to address what does an extension or another grower advisor what would people on the call recommend that they say to growers who come to them with questions, or what you recommend that they know just as they go about their daily business, maybe even designing their experiments. So Jozsef, after hearing all of the conversation so far, what would you recommend to, to me, to other extension people on the call, to people who work with growers about knowing, the existing farm situation, the management history, the use of other materials about, say the mycorrhizal products that you're very familiar with? What would you recommend that they know based on your experience?
Mm-hmm. Well, Matt, I would like to go back to the original conversation that we started up with. Is where is the data? So I have a couple of comments on it. That actually there's a lot of data out there. There's been a lot of publications out there, but certainly most of the information that are available, on these microbial inoculants and microbial fertilizers are not in a public domain because, companies generate a lot of data, and certainly I can speak for Mycorrhizal Applications and Lyon Biosciences only, but what I know is that we generate tremendous amount of data to support, product sales. So primarily we get the response or the feedback from growers, what their need is, and we try to set up many trials every year. Unfortunately, this is a company environment. This is not an academia. So even though we generate tremendous amount of data, and most of these trials are under controlled conditions and done by either our internal researchers, contract researchers, or even university researchers. We are, within the company, we are not rewarded for putting information out there in a form of publication. So, I used to work at academia as a researcher, and I published quite a bit because, I was my performance was evaluated based on, how many publications you put out there. But, in industry, there's a different situation. So that's kind of a problem that, even though we generate a lot of data, we just don't make it public because we just don't have time. We are not rewarded, for publishing in scientific papers. Even though, these are company-sponsored studies, we really have to be careful, how we generate the data because, certainly you can hear from growers that, companies have their own data. They can say whatever they want. They can cherry pick the data and show us, the best results. But, you have to keep in mind that to be able to register a product, you have to go through a multiple year project, which is product development, where you have to generate enough data to convince senior management and the whole company to register the product. That's a significant cost to the company. So we have to make sure that we have enough non-biased data that the product really works. Well, certainly some of the companies just don't make their own product development. Some smaller companies, which I understand, they don't have enough data. But, larger manufacturers pay quite a bit of attention on making sure that, what you put on the market that really works. And certainly there are, in particular in microbials and soil inoculants, there are some limitations. And,
so Jozsef, hold on, if it's okay.
Yeah.
Please just, you don't have to play along, but just hear me out. I would, a cynic could say that the rise in the value of the microbials industry indicates that data are not needed to make money at it. But there is some, and I don't want to call it pushback, but there are some genuine concerns about the longevity of the use in the absence of public domain data that is verified and verifiable in the traditional ways. Because at some level, the microbials industry, at least in my experience, stands apart from other inputs at some level in not being represented in the traditional channels that many people rely on. So you can go to conferences and conventions and workshops and trainings and this and that and the other thing. You do see reports about products that are essentially competitors with these products. And they have independent third-party testing and or company data that are somehow that are presented in an open forum. And now I'm probably not traveling in the right circles, and I don't want to overstep or misspeak, but this doesn't seem to be the case with a lot of microbial biostimulants.
Yes, I totally agree with you, Matt, because biostimulants and microbials, plant hormones are different than regular, let's say, hard chemicals that is easy to test. Even within our internal and contract researchers, it's really hard for them it was hard for them to understand that these are not fungicide. This is not just to spray and see, the product work, yes or no. It's, you really have to consider the environmental conditions, the soil conditions, the moisture content, the host plant. It's a very complex situation.
I totally agree. I totally agree. I'm interrupting. I'll just be a voice, and we need other people on the line to interrupt me for sure. I would say the microbials industry at some level is getting a quasi-free pass when they say it's complex. Because I can point to a lot of research on other inputs that's equally complex in its own way. And nobody runs from the complexity of that work. They address it head on, however flawed it might be, they address it head on. They don't wave and say, well, it's complex. It's mysterious and you have to have 95 contingencies in place to have a valid trial. If that is the case, I just wonder openly about, the typical response that a farmer will get on a typical farm where those 95 contingencies are not in place. So in the third call, we'll get, that would set aside to address, a possible way forward for people who are interested in such a thing, for a different type of testing program. But, here today we're talking about the take-home messages from the on-farm research, and maybe we need to hit a pause button and just say, if Steve, Dennis, Joseph, and Philippe, if you want to offer a take-home message, what would it be?
Well, Matt, I want to comment on this, that it's very complex, but I think these microbial inoculants and biostimulants are relatively new to the market, even though mycorrhizae have been produced for decades. But there's a sharp increase in number of products on the market, and it's kind of new to everybody, and it just takes time to understand how they work. For example, when I had a technical meeting earlier this year, and I presented data that, let's say, we had a 45% increase in production in lemon under salinity conditions. So when you made a presentation, you really have to emphasize that these data are valid for particular conditions, for under stressful conditions. And you cannot interpret the data because, some people just, say, let's read the summary and, they got 55 or, 40-50% increase in yield. Let's try this product. But since these are, particularly I'm talking about mycorrhizae primarily and mycorrhizal inoculants, these products are developed to counteract stressful conditions. So if the production conditions are optimal, you have less chance of getting the benefits out of these products. I'm not saying that it's not impossible, but that's just harder, and you would see less response under optimal production conditions. These products, what I'm talking about, don't really give you a very strong effect when the growing conditions are optimal and the plant produces under optimal conditions. So that's what we have to keep in mind, that most of these products are developed for mitigation of stress. And if you have a large grower who pays very much attention to putting the right amount of fertilizers, the right amount of water, everything in place, and everything is optimal, it's definitely harder to show the benefits.
Fair enough.
I would like to add something there to the subject, if it's possible.
Yes, please do. Do.
Yeah, okay. With bacteria, you see a very wide, diverse mechanism of actions that are acting on the crops and on the soil. Furthermore than with mycorrhiza, probably. And the thing is this, that Even, for example, I had worked a lot in the flower industry where you have in South America, it's very technological. Every bed is controlled with a computer in some places. It's very, very calculated. Even under those conditions, we can still get improvement and see the improvements, even though they are under fantastic conditions. However, of course, the magnitude, as it was pointed out, the worse the conditions, the better the effect you can see. Now, the point that I want to make is that sometimes with the microbes, we are acting in such a different mechanism of actions that you might be looking at different factors. It's not only yield that you can increase. You might get an increase of 10% yield or 5% yield, which is a very low compared for an interested person. But then you look at quality, for example, the quality, the sugar content of your fruits, the shelf life of your fruits, all these factors that will be improving but not being measured because it depends on the type of crop, and the user. Then that complicates a little bit the research because then you have a multitude of factors combining crop type, soil type, and variables that you want to look at. It's not like a fungicide that you just look, okay, this is the fungus controlled, yes or no. It's a very quick result and very quick observation. Here, you have a whole array of factors that you want to look at and that might benefit you simultaneously, and in different proportions.
Good points.
Actually, I totally agree with you. And in some cases, I could add that we are looking at, potentially, reaching the same yield as optimal conditions, but we could reduce the amount of water by 30% and, the phosphorus fertilizers. So these are also additional things that, not everybody looks at.
Oh yeah, absolutely. So Dennis and Steve, we're offering some take-home messages, I guess, for everyone on the call that you've gleaned from your very large amount of experience with the on-farm testing and trialing. So far, I've heard, and please correct me if I'm wrong, I've heard that they may be best under less than ideal conditions, the products that is, may perform best under less than ideal conditions. They may affect factors other than yield, for example, quality. The effects will differ by product type, for example, bacteria versus fungi, or mixed product. Dennis, Steve, what else would you like to offer to that list?
Hey Matt, this is Karthik. Can I jump in for a minute?
Yeah, please do. Yeah.
Yeah. So apart from all this, I think some of the challenges what we manufacturers face is unless we have a study done by an educational institute or there is some public available data, we cannot make claims on the product labels itself that are not supported. Or there are strict guidelines in that area.
So where does that leave you? What would you recommend as a possible next step, or what would you benefit from having? Who would you reach out to for help, or how could we make things better?
Well, I would say, if you have some data during this whole exercise that you could publish, that could be a big help to the community itself.
So I want to make sure I understand correctly. Would you look to rely on the research extension community to develop those data?
Well, not necessarily. If that helps the community as such, really, to it could go both ways. That it provides enough information to the farmer or the grower to look at and then see what individual bacteria is doing, as well as it also provides some support data for the manufacturers like us to make those claims on the product itself.
Sure, it's my understanding, and I could be off base, but it's my understanding that the amount of literature on modes of action and effects under controlled conditions, semi-controlled conditions, like a research station, greenhouse kind of setting, that the amount of information in the refereed literature of that type far exceeds, orders of magnitude exceeds the amount of publications that farmers and farmer advisors would be able to apply directly day to day. in terms of efficacy, product selection, product handling, product, everything that I think is important to people on the call and people they work with. Is that a reasonable assessment?
Sure. Yeah, definitely. There is literature out there, even if you can find information on the public domain, you cannot make those claims on the product itself because, it's not coming from a reliable source.
Fair enough. So,
I would like to add something that I think is key at this point, if you don't mind.
Yeah, please.
One thing is that, for example, we run experiments with universities. They are very expensive and they are very specific. And a lot of growers will look at that and say, oh, yeah, that work over there and with that crop, doesn't apply that much to me, On the other hand, for example, my experience was, for example, in California, growers really accept data or information provided by the extension agents because they are kind of the connection between science and field. And they have a broad view of users that have used the product under different conditions similar to the ones that they have used their product. Then there is a very good factor of credibility on those extension agents, that have built a reputation in the field so that people will go and say, okay, I want to try that product.
Excellent point. I would agree with that observation. I think part of what has drawn this team together, and I want to just emphasize that Philippe and Jozsef and the Tainio group and others are the first companies for people who don't know this, the first companies who stepped forward when Bonnie and I brought this idea to the larger community, and that's clearly obviously one reason why they're on the call and taking the time to do that and sharing their opinions. And we mentioned on January 24th that there are something in the neighborhood of 177 products that have the 4 characteristics I mentioned up front, and that's spread across in the neighborhood of 60 or so manufacturers. So we have what could be considered a normal bell-shaped curve with regard to company interest in developing the kind of resources that will really help people, use their products responsibly and effectively. And again, I'm not making the pitch for the companies represented here. I'm just mentioning facts. Okay, they were very receptive to having this kind of dialogue. Okay, and that's to their credit. So we are focusing on the on-farm research that people on the call have done. Jozsef, if I could go back to you, if you wouldn't mind, just in a real quick, summation, a minute or two, could you give folks on the call a sense for what your average research trialing program involves, like maybe on an annual basis, like number of sites, number of growers, number of products? I mean, without divulging trade secrets, just what does it look like structurally?
Yeah, so, I have to give you a bit of a background on this, just very briefly. So, Lyon Biosciences, that I used to work for, acquired Mycorrhizal Applications, and that company was a US-based company. So, our goal in the next few years to expand this business throughout the world. And so, we are focusing primarily on foreign countries and also supporting the U.S. business as well. For this year, we're going to have nearly 200 trials in several countries of the world, and we are focusing primarily on those crops that are important for particular regions. So we cannot just, determine that, okay, so let's say corn is the largest, corn has the largest growing area, and okay, let's go to corn, because, if the product doesn't work in corn or whatever in other crops, it wouldn't make sense. So we basically start from, our starting point is the grower need or the need for solving the problem, and also, evaluating what kind of stress situations are out there. What is the largest stress affected area, what are the largest, soil conditions in these growing areas. So these are the main factors that we consider when setting up a trial. And we have cooperators. We do internal trials. We have cooperators with contract researchers. So they follow the protocols, and they have in several countries, they have strict regulations how they conduct the trial. And also we work with university people because, some of the growers are they would like to be, so they would like to see data that were generated by independent researchers, which is totally understandable. And we, in fact, like to have these kind of backups that, in fact, we could use published literature as well, since university folks like to publish, their results. So basically, we work different kinds of people to generate data. And as I said, unfortunately, most of these data are not on a public domain. So what I could recommend is, if a grower is kind of in need of, or extension person is in need of particular data, I think the best is to contact the company, and the company would be happy to provide you with the data, or just talk to the technical person, and get some information from them, and ask them, when they attend a meeting, when they host a technical training, a seminar or something. I think that would be a good source of information.
Sure.
To get these non-published data.
Right. So what I heard, if I could interject, what I heard is a multinational. The structure of your program is multinational. It includes the U.S. but other countries. It's on high priority crops. Crops are chosen to represent regions, regionally significant crops. Presumably the products as well is a good match because of what you were saying earlier. It has to be vetted. There has to be sufficient evidence that it will work. You have your own cooperators, and you also work with university people, and all the data funnel back to the, if you will, the corporate office for analysis.
Yes.
It may not get into the public domain directly, but growers and people who advise them could always.
And Matt, the other thing that I have to mention,
call
even
you
though,
up.
These trials are conducted in replicated large field trials. Some of these data sets are not enough it's not really sufficient to publish in a scientific journal, because for a paper you have to look at different aspects of you have to look at photosynthesis, and you would like to back up the yield data, but sometimes we just look at the yield, the reduction of fertilizer, reduction of water, and that's all.
Sure, that's fair enough. We don't. I think that's to be quite honest, personally, that's a larger topic. Okay, I think there would be I think there are journals who would actually welcome, yield and basic plant growth data that are possibly may being collected. Okay. But that's a subject for another time. Philippe, I know that you set up and you manage an extensive trialing system also. What are the characteristics that in a site or a cooperator that you tend to look for as being most productive?
Well, I work a lot with growers and a different scale of production. I'm looking for people that are curious, especially, that they are not just into making the money, but knowing what they are doing and to improve it, to get, so because these people will provide you, you will have more feedback from these people, yeah? And that's very important so that you can always go and modify things. Usually, the problem is that you have a specific protocol that you suggest. For example, you're running a university, a protocol, and it's fun and dandy, but doesn't mean that protocol can be applied in a farm, because they have their own management techniques, they have their own, system going. Then it's going to have to be always applied to a system. And that's when you have to be very careful about seeing, okay, how do you do the application? For example, I work a lot also in bioremediation of soils. We come sometimes to some farms and do the bioremediation first to destroy all the pesticides that are remaining in those soils. Then we can come and we get an amazing success compared with those fields that have not been bioremediated that were treated with the same chemicals. Yeah, the same way you can use combine, because these people are using, let's say, copper or things like that. You can chelate copper with silica, for example. Then you can restore those soils, even they have been soaked with copper. You can bind it, chelate it completely, and then have a complete success in the growth of these crops. Yeah. Also, the frequency of the application of the product, the sequence of application of the products, all that stuff, all those programs, you want to see them, and that's why you want a good participation and communication with the growers so that you have a good understanding and feedback so that everything, goes for the education of both parties.
Thank you. That's, I jotted down curious, and I think that's, to me, that's an incredibly important word that you use there. I mean, somebody who's right from the beginning interested in possibly doing something new or different or looking at their system differently. Dennis or Steve, as far as your trialing program and your evaluation program, what would you recommend to people either listening now or who might listen after the call as a grower or a grower advisor? What would you advise them to do as they consider your products in terms of trialing? Would they contact you directly? What are the generalities in terms of setting up a trial involving your products that you would look for?
Generally, in working with growers, it comes similar to what has been talked about is a mindset that is focusing on wanting to make a change in their agriculture production. The other thing I look at is there's a clear definition of what we are trying to do. So generally, what we will do is sit down with the grower and have a protocol sheet, which we will fill out based on what the definition of exactly what we actually need within this trial. Because I think that's very important that we all have a clear focus of where we are starting and where we're going to head. And then it goes back to education, understanding that we get all of the information of what they are going to be doing in a farm setting and how that can or can't affect the outcomes of that test that we're going to go ahead and do. And generally, we recommend that they be some kind of control so we have some way to monitor what the actual, rather than just visuals, or rather than just saying, this is what we ended up with, we have a visual difference of a control and a trial within the existing field, which is usually what we like to see.
Yeah, we're generally very open to conducting trials. We work with quite a few growers, universities, and people throughout the year on a year-to-year basis. And in general, Dennis and I will talk to people, and it becomes fairly apparent fairly quickly, usually, if the person's after free products, that's usually not the situation that we want to go after. We typically don't pursue those as much. The term curiosity is one that I would say definitely piques our interest. If we're trying to find out something new, trying to apply to something new, a different crop, a different scenario, if we have people that are going after a specific problem, well, I'm applying a lot of phosphorus, I really want to apply less phosphorus, well, let's see what we can do. Or if I want to reduce my nitrogen applications and I want to improve my efficacy of the nitrogen that I apply, what can we do there? So we'll go after, sometimes with growers, it's specific problems. Sometimes we'll have growers that, I want to get my sugar up. I want to get my yield up. I want to get my storability up. So a lot of times it's simply coming to us with an idea. Well, what do we want to do? What is the approach? Tell us more. And we keep coming back to the idea of the ER and the patient, but a lot of times the field history makes a big difference. So we need a bit of information, a bit of data about what's been going on. And as Dennis said, what are the goals? What do we hope to see? What do we hope to achieve? So our approach is curiosity and hopefully to solve a problem that somebody's been having. And mentioned before the idea of seeing the most benefits in the worst situations. And I'll be honest, sometimes when we're working with someone new, our approach is give us and let us see some of your worst area. Because a lot of times to see the easiest benefit, the visual benefit, that's where we can go. So if we have somebody who has a dead spot in their field, well, let's try to figure out why, let's remediate that, and let's move forward with that. If I have grass that's not establishing or something like that. So sometimes it's those sorts of tests and trials as well as just taking somebody who was completely failing, trying to figure out why they were failing, and just moving them to a situation where they can actually produce and grow something.
It seems to be a very, there are obviously some common threads, not only across the people we're hearing from, but also, Dennis and Stephen, your approach to various people, common threads, but it's a case-by-case basis, it sounds like. I have a lot to learn in moderating these calls, and one of the most important things to learn is let other people talk and not let me run ahead with my questions. We have other people on the line. I want to just make it wide open for people to speak up and ask say or ask whatever they would like to along the lines of the topics that we're addressing. So I'm just going to be quiet until somebody speaks up.
Hi, Matt. Dave Benshoff here. Can you hear me?
Perfectly, yeah. Thanks.
Okay. Something that came up earlier was a question about broad-spectrum fungicides and bactericides could possibly work against your microbes that you're trying to build in the soil. And products that are commonly used by organic growers, such as myself, are copper hydroxide and hydrogen peroxide. But these are normally done in a foliar application. So I guess the question to ponder would be what kind of an impact is that going to have on microbes in the soil? Is that really going to be a problem?
I think we mentioned, this is Steve weighing in, and I'm sure others will jump in as well. The mode of action of the material you're using is going to make a significant difference. What is that material? If you have an element like copper that's not going to be broken down by the sun very readily, it's not going to react with organic like your hydrogen peroxide will and basically dissipate fairly quickly over time. If you have a long-term, fairly resilient material like an elemental approach, coppers, sulfurs, those sorts, those are typically going to have a longer effect. And we do, as was mentioned before, we have to chelate those, we have to pull those out of the physical system. Many of the pesticides, herbicides that are used have a relatively short half-life, and a lot of times those are either going to be photolysed and broken down by the sun if it's applied through the foliar, or if it does wash down in the soil system. There are many different bacteria and fungi that can break those materials down, and they'll simply use those as a food source. I mean, 2,4-D is an example that's not really used in an organic setting, but many of the bacteria and fungi species they're quite happy to break that down and use it as a carbon source. So it's going to be kind of mode of action is going to be critical. If I have a bactericide or a fungicide that is very, very broad spectrum in its activity, in that if I have a fungicide that is specifically going after a specific metabolic function versus one that's broad spectrum, say it's breaking down the fungi's ability to produce chitin, or if I have a protein synthesis inhibitor that pretty much every organism is going through and using those, like glyphosate in your shikimate pathway, those broader spectrum are going to have a bigger impact. So when we look at these, we have to know a little bit more about what they are, what their half-life is, and what their long-term fate and metabolic byproducts are going to be. So hydrogen peroxide is an example, breaks down readily, dissipates quickly. Copper is an example. You're going to have a longer term, and you can have some severe accumulation problems with those elemental types. And chemistry, chemicals, you're kind of everywhere in between. Some are readily broken down very, very quickly, and others take a bit longer.
You know, one thing.
Yes, and that would explain why copper is a restricted product under the organic rules, and you're not supposed to use it 2 years in a row in the same place.
I'm sorry.
Yeah, we'll let Dave. We'll let Dave finish out.
It has to do with the health that we start with based on our soil environment. If you're an organic grower, the other thing that is very key to that is the concentration rate and also how often you're using it. From my experience, the biology can bounce back in some instances relatively quickly on a use that is a one-time use in order to control a problem, especially if we go back there and do something to either re-inoculate or to feed that biology. But continued applications over a very short period of times, and I'm talking high concentrations on 7, 14, 21-day intervals, is over repeated applications, as I see, sometimes more detrimental to the biology.
Other questions, comments?
I'd just like to weigh in here and say those were a couple of really great answers.
Bonnie, what makes them stand out? Just so people get from it what you would like them to, for sure.
I guess pointing out the fact that it is somewhat complex. You have to pay attention to application, concentration, and this thing about the question of if you're applying things on the plant, how does that affect microorganisms in soil? We need to be aware of the fact that these microorganisms, whereas they spend part of their life in the soil, they may also spend part of their life inside the plant or on the surface of the plant.
That's an answer I was looking for.
It's a dynamic world.
That answers my question.
I would. This is Andy in Rhode Island. I'd just like to mention that copper in soils that have appreciable organic matter, copper is very tightly bound. So on an organic farm, which presumably where someone is working to build up their organic matter levels, what copper you may be applying foliarly to your plants is probably going to be held pretty strongly and may not have a tremendous effect on microbial activity.
Yeah, especially if you remove the plant fodder at the end of the season.
This is Steve in Cincinnati. We're going to move off from that subject. I've just been looking at the database that OSU's put together on their website for the products, and this is really helpful. I'm a new person, but just to be able to see the species that are in the products, and some of them have 3 or 4, and some have 10 or 15. But just to see it all listed out, and the other thing that strikes me here is the number of things that say to be determined in terms of shelf life and mode of action, and third-party verification. Almost all are blank. But just to have all the information in one place is helping me wrap my head around it. So I appreciate that, and I appreciate Matt's work to try to get some more data and research out there. And the other thing would be what specifically are these products aimed to do? And that's not listed on here at all. So I've got a lot to learn, but this is very helpful.
Appreciate your comments, Steve. We're trying to improve that interface all the time. We're getting input that helps us with that, so we like all input, pro and con. I know that we can adjust it, but the idea is to put as much information in one place as possible, as is reasonable. So any other comments or questions for people on the call, whether they be fellow callers or resource people?
Yeah, I have. Okay, please go aheed.
Yeah, this is Mark Schulbeck. I was just looking at a catalog of organic products. The things I'm curious about are biofungicides, things like Serenade, which is based on Bacillus subtilis, something called double nickel biofungicide, and that's a Bacillus amyloquapians, and then I might have mispronounced that. And then there is another fungus, a root shield, which is based on various trichodermas, a couple of strains of trichodermas. I was wondering if any of these that are antagonistic to fungal pathogens could also turn around and attack desirable fungi that either are present in the soil or that you may be applying in the form of one of these products.
This is Bonnie. There are some incompatibilities that we've been aware of as far as the biopesticides themselves. Say, for example, a trichoderma and a Pseudomonas. If the mode of action has to do with chelation of iron, then the Pseudomonas wins that battle and robs the trichoderma of iron and will reduce its efficacy. So there are these things that go on. I mean, they're living organisms, and they compete with one another.
Mm-hmm.
Actually, this is Andy in Rhode Island. That's very related to what I wanted to ask about in general. Is what is known or what's been studied about antagonism between native existing soil microorganisms and those that are contained in these products that are being applied?
Well, if I may intercept here, I did some interesting work for years in looking at the soils and the biology of soils, and we were adding a very minute amount of bacillus to these soils and other strains, and compared with the huge populations of microbes that were there, they were very, very small. What we noticed is that these microbes, these bacteria that we are adding to the system, maybe with time they will disappear. We won't see them anymore. But there was a complete shift in the communities that were present in the system. They were more stable. You increase nitrogen fixation, you increase beneficial fungi, you increase aerobic bacteria, you reduce anaerobic bacteria. There was kind of a modulation of the system toward a more beneficial system. Yeah. Then yes, what I'm trying to say is that these some of these bacteria that are widely used, these beneficial bacteria, they are not going to be there forever. Yeah, because they form part of the food chain. They end up being eaten, consumed. You know, they may not establish well because they are not their natural system. That's why you have to reapply them sometimes. You know, to keep them going because they will be consumed by the system. But they tend to have a beneficial effect as far as the community by triggering by it's like a jump start into a more efficient system.
Mm-hmm. Okay, thank you.
I think somebody else was going to jump in with a question or comment as well. Now would be a good time.
Well, this is Steve again. This was supposed to be focused on the biostimulants and not so much the biopesticides, but a lot of the organisms are the same ones. And does that cause any interesting side effects, or does it cause you problems to try to compartmentalize them when they're the same species being used in both places?
Steve, if that's a question...
This is Steve here. That's something that we've historically seen many, many times. In order to, and this is something that we discussed the last call, so you might listen to the recording a little bit, but the long and short of it is, to be identified as a biopesticide, to have control claims, you have to be registered through the EPA. And that's kind of the hurdle that has to be overcome in those situations. So do you see those organisms, similar organisms? Well, yeah. We see benefits associated with bacillus. A lot of the bacillus subtilis, amylophacians, pumilus, and others, they'll both have biofungicidal properties as well as phosphorus solubilization properties. They'll be indole acetic acid producers or other phytohormone producers. So people like to put these organisms inside of boxes, but just like a tree grows, it's not just producing a fruit for me. It's producing leaves. It's producing exudates that go out into the soil. These bacteria are doing the same thing. They're living their life, and sometimes down there in the soil and on the leaf surface, life gets really hard. So all of these organisms basically have to. They're like the Boy Scouts. They've got to be prepared. They have protective capacities to themselves. They'll produce bactericides, fungicides. They'll be ready to deal with biotic and abiotic stresses. If your salt content starts to be too high, electroconductivity is too high, or you start to encounter a drought scenario, these organisms have the capacity to sense the environment around them and react to the environment around them. Something that I'm really excited to start to see more of is research into plant exudates and how those exudates are specifically being produced by the plant to feed specific organisms. We're seeing that in the exudates from the leaf tissue as well as out of the roots. So the plant's microbial interactions, we're just starting to get into an understanding of them. But I am excited because it's kind of a brave new horizon of what we'll see, because the overall interactions, I don't think we fully understand yet. We're starting to grasp the genetic ramifications of these organisms. Share plasmids, they share DNA. So if there's a stress environment and a protective plasmid is produced, that can be spit out in the environment. And so an organism that was susceptible to a specific antibiotic can pick up a plasmid and now be protected from it. So these are very fluid organisms in a very fluid environment. So there's no single compartment for them. These like to shift. These are living systems, and they are constantly changing, moving, and mutating.
Thanks. That's helpful. Thank you.
Well, I would like to add something there that might be interesting from the scientific point of view. These bacteria, for example, let's take Bacillus subtilis. It can produce a huge array of antibiotics, and if you look at the literature, you might find more than 50 different antibiotics produced by Bacillus subtilis. Now, they can do a lot of different mechanisms of action, as it was mentioned before, and what's interesting is that even though they have the capacity to produce those compounds, they are not producing them all the time, because they will be completely burned out by producing all these compounds if they are not going to be of a purpose. Then those compounds are produced by a stimulation from something. Then when they have the need of producing something, they will do it if they have all the building blocks. That's why we don't have to be very afraid of putting these quotation mark biopesticides in the soil, because they are not going to be factories of toxic antibiotics all the time. They're going to be modulating their activity depending on what they are doing.
That's a good point. And when they do produce them, it's very it's at that microsite. You know, if it's up against the root or wherever, so it's not in huge volumes that these compounds are produced.
Exactly.
Folks, I'm taking a look at the clock, and as always, we have a whole lot more to talk about than what we have time for, which actually I think is a very good sign. And I just want to thank everyone again for offering a great meal. This has been very, very helpful. We do have a few 15 minutes or so left in that neighborhood of 10, 12 minutes anyway. I want to make sure that if there's a topic or a question that somebody has not been addressed so far, that anyone on the call, anyone at all, thinks they would really feel left out, if it wasn't addressed, that now would be the time to bring that up. Okay, hearing silence, that would be. I'll do that.
Well, I want to add something. I want to add something that I think is important, because it's an observation that I get from a lot of growers, and they say, okay, they think that by applying a microbial inoculant, they could eliminate completely their fertilization program, for example, and things like that. Do go to very drastic approaches, and that's never the case. You always have to feed the system in some way or another. Maybe you can reduce the amount of fertilization, maybe the amount of water, because we have seen reductions in 40% of amount of water needed, 35 to 40% in fertilizer, but you always need some, Then I think that's something that has to be very clear for the people to understand.
Well said, and very important to note, definitely. Anything else from the group that they need to bring up or ask?
This is Mark Schoenbeck. I want to pick up quickly on my earlier question. If I were a grower, an organic grower, and I really wanted to rely on microbials both for managing disease and for these direct biostimulant, biofertilizer effects, and maybe I'll direct this to the representatives of Tainio and Ecomicrobials and Mycorrhizal Applications. If I am using, let's say, a trichoderma product or a Bacillus subtilis, Bacillus pumilis product as a biofungicide, would you recommend, if I was also using one of your products to improve plant performance and overall soil health or reduce stress, would you recommend I put them on at different times? And if so, how much time would I leave between them? Or would you strongly recommend I consider not using one of the biofungicides?
I work a lot in combinations of microbes. I study a lot the interactions between microbes, and definitively they don't perform additively like we would expect. For example, you have two microbes that both control a disease in 20%. You think that if you put them together, you get 40. Sometimes you get 0. Right. Then that's my recommendation is, since you haven't done the work in the lab to see if there is an antagonism between these organisms, I would really space out the applications if you want to use all these products.
Yeah.
And we generally agree. We like to see a food source put out with our microbes, but anything that's being used as a control, we like to see space in between. And quite honestly, typically the more space, the better. We usually like to see a few days, 3-4 days, so you can start to have the activity of the product you're applying for control. And then you separate that out from the product you're trying to use for stimulation. So with our products, we like to see separation, 3 days, 7 days is usually better. And then we like to see some fish, some humate, some other carbon-based fertilizer applied with those microbes to help feed them and help them establish with the plant until they can start being fed the exudates by the plant.
All right. Thank you.
Good questions. Any others? Okay. I also would like to put a question to the manufacturers on the call. I know that most of what I'm hearing is context specific. That very broad statements don't always apply. We've condensed a lot of those individual comments into some broad statements, but I'd like to go back to that one more time, if you will let me, and just ask if you could name one thing, one recommendation, just within the confines of this call, right, that people are listening to now and they may listen to later, and of course they may follow up with you on, but if you could name one step, give them one recommendation, if they are a grower who is new to microbials, biostimulants, what would you recommend that they do to start off well?
Well, there's so many factors that you could see that are very important. You have to see what products what are you looking for, in what are your needs of your system, and to see what type of product will bring this into the system, how you apply it, how you bring it into the system. The form of application is very key about the success of these products. They have to be localized. They come with the irrigation system. They have to be applied directly to the seed. All these concerns are very important because you can flood the system with microbes. That doesn't mean that they're going to get into the target place. For example, mycorrhiza, you really need to get it to the roots. You can have a lot of mycorrhiza in the soil. If the roots don't get to that area of the soil, those are wasted. Things like that. Also, when do you do the application? The time you don't want to do it in the full sun. You do it in a foliar application because they're going to be very stressed, and the irradiation of the sun might affect them very much. Then try to do them at the end of the day or early in the morning. Then those are the type of things that is very important considerations. The way of applying the products as well as what product you're going to use.
Thank you. Well said.
I'd like to weigh in, and I agree with all of that. I love the old proverb of the most important fertilizer, your footprints in the field. And basically that boils down to understanding and observing what's going on in your field. Many of the problems that people run into can be avoided through observation and management. So having a better understanding of what's going on in your field is going to be the key to making sure you can make these products work.
Okay. Joseph, anything to add?
This is Dennis.
Well,
I would agree with
to
all
best
that.
start, I would say do your research. I mean, look around, do your research, ask company people, ask other growers who have tried what their conditions were, and try to relate it to your specific conditions. Because, as we stated earlier in this call, these are not very easy things to determine, and these are very specific compounds that work under special circumstances, and you cannot just buy them online and apply them. So I would say first, before even starting applying or buying a product, do your research, try to find out what your problem is, what you want to address, what your problem that you want to solve, and just do your research.
Okay. I'm sorry. I cut somebody off before Joseph.
No, actually what Joseph just said, this is Dennis. I was going to basically repeat exactly what he just said. The other understanding is that I tell growers all the time is generally a microbial product by itself is not going to fix a problem, as we talked about earlier, on a silver bullet. And so a lot of times, based on doing your research, also get an understanding of how are we going to keep that biology alive. Steve had mentioned earlier pertaining to making sure we supply a food source with that biology. Some of your carbon products can actually protect that biology a little bit, as we were talking about during the day and temperatures. If it's a soil-applied product, it is important to understand when is the best time to apply it, incorporation, root dip, soil drench, what is going to be the most effective way for you to implement that product into your program in a way that we have put it there so it is alive and functioning, so it has the best chance in order to be successful. And I think some of that has to come down from information, that you do your research on the products, and then you make sure that you're getting all the information from whoever the rep is or the company that you're dealing with, and make sure they're also, we put a lot of good questions on this on this phone call today. Make sure you're asking them those questions pertaining to your use and whether or not they have the understanding of the best way to use that. If they don't, then I would probably look somewhere else for my biological products.
Okay. Very well said. Very, very helpful. I just want to thank again everyone.
May I add something?
Please
very
do.
Quick?
Yeah.
May I add
please
something
do.
very quick?
Yeah.
Please don't try to improvise. Usually, you might get very good ideas, but it's good if you run them by the person that is selling you the product, the manufacturer, something like that. They might tell you, no, that's not a very good approach. Somebody tried and failed. Or this is the reason why you should not do it, or do it this better way, But it's always good to consult.
Yeah, and one last thing, I guess, is one of the things I tell my growers all the time is this is not a product, it is a program. And we have to buy into a program in order to be successful at establishing and getting results with biological products.
It's fantastic. I feel like we're starting the conference call 3, which is no, seriously, which is exactly what we could hope for, at some level, right? Speaking of conference call 3, before we end this one, it's on March 10th. It's at the same time as this one, 2:30 p.m. to 4 p.m. Eastern Standard Time. The call-in connections will be, I think, identical, but you will get, if a person registers, they will get the connection information. So far committed as resource people, we have Brian McSpadden Gardner with Sustain. He'll be back with us. We have Bruce Caldwell from 3 Bar Biologicals. And then we have some members of this program team, including Carol Goland of OFA, the Ohio Ecological Food and Farm Association, Bonnie, who was on the call today from the University of Tennessee, Sabu Kumarapan, who is a colleague of mine here at Ohio State, but he as an ag economist. Those are 3 people who have committed to being resource people, but as I mentioned, we all learn from each other in different ways. So please do encourage people that you think will find these calls to be useful to register for them. Registration obviously is very easy. It just helps us to connect with them. So if you have any questions about that, obviously contact me or Jung. But again, I want to thank everyone who called in, especially our resource people who committed their time. I think it's been a fantastic conversation. We do plan to post the recording if our recording system worked well. We never know until we try it. But we'll post the recording, and we look forward to hearing from you again. And as always, if you have questions or comments about what you're hearing or not hearing, please do bring them up. We're interested in all input. But unless there are further comments or questions, I think we can wrap up for today.
Thank you, everyone.
Thank you.
Thank you.
Thank you, everybody.
March 10th is a Friday.
Thank you.
Thank you, everybody.